and today we will read about plasma membrane. So plasma membrane, as we know, it encloses every type of cell and both prokaryotic and eukaryotic. It physically separates the cytoplasm from the surrounding cellular environment, and its size is almost 8 to 10 micrometer. It is elastic, living, dynamic and selectively permeable, means it does not allow all the substances, it allows particular type of substances to come in or come out of the cell. Now, as it controls the exit and entry of molecules ions, this generates differences in ion concentration between interior and exterior of the cell. Before going to the detailed part of plasma membrane, we will first cover how it was discovered. So we will see the history part of plasma membrane. The term plasma membrane or cell membrane was given by C. Nageli and C. Karmer in 1855. Now in 1895, Charles Overton suggested that plasma membrane is composed of lipids. So basically, as it is a layer, it is composed of lipids. Then in 1900 to 1920, further studies were done on plasma membrane, which stated that lipid must be a type of phospholipid. Then in 1925, further studies were done by E. Gorter and G. Grendel, who proposed that plasma membrane is composed of phospholipid bilayer. So we see in 1925, E. Gorter and G. Grendel propose that it is a type of phospholipid layer. Next, we will see what is the chemical composition of this layer. Here you can see a picture of plasma membrane. It is composed mainly of three components. So we will see the chemical composition of plasma membrane. So mainly it is composed of three components, which is lipids, proteins and carbohydrates. Now we will see that in a human normal RBC cell, RBC of human generally has a plasma membrane which has 52% of protein in it, lipids are 40% and carbs are 8% in normal RBC of human plasma membrane. Now we will see that here you can see the inner face of plasma membrane and the outer face of plasma membrane. On outer face you can see sugar side chains and cholesterol in the inner face, integral or intrinsic proteins on the inner face which are present in embedded inside the membrane, and peripheral or extrinsic proteins which are not embedded in the membrane but can be present outer or on the inner surface of the membrane. Then here hydrophilic and hydrophobic part of layer is shown. The hydrophilic means water loving, means the head of phospholipids are present on the outer side of the membrane, and the hydrophobic water fearing fatty acid tail of phospholipid is present embedded inside the membrane. Now, as we have seen the chemical composition till now, lipids, proteins and carbs, we will now see what types of lipids are present inside the membrane. So classifying the lipids, there are 4 major classes of lipids. 4 classes of lipids are present. One is that we commonly see and present in majority on the plasma membrane is a phospholipid, most abundant lipid. Then sphingolipids, and the third type glycolipids, and last sterols that we commonly call as cholesterol, for example. The type of sterol present inside the plasma membrane is a sterol. Now all 4 classes of lipids are amphipathic molecules possessing both the hydrophobic and hydrophilic domains. They have both hydrophilic part and hydrophobic part. How? As you can see in this diagram, a phospholipid is shown which is amphipathic, means it has a hydrophilic head and also a hydrophobic acid tail, water loving and water hating part both are there, so it's a amphipathic phospholipid. Then we will see the proteins part, which are 52% inside the membrane. So if we see proteins present, then we find that 2 types of proteins are present inside the plasma membrane. First are the integral proteins or intrinsic proteins that we found as embedded inside the membrane. These are responsible to span the membrane, means the movement of membrane, the flexibility of the membrane is dependent on these type of proteins. Next is peripheral or extrinsic proteins. Now these are present on one side or the other side of the membrane. These are not present embedded. Either of the side these are present on the membrane. So proteins we have found, for example, this is the membrane. So those proteins which are present inside, embedded inside the membrane, these are intrinsic proteins. And those which are present on either side of the membrane, these are extrinsic proteins. Last, we saw the third type of component, which was carbohydrates. Now carbohydrates are either short, unbranched or branched chain of sugar attached either to exterior ectoproteins that forms a glycoprotein, here you can see in this diagram, or to the polar ends of phospholipids at the external surface of plasma membrane, which form glycolipids. So glycolipids here in diagram you see only on extracellular side, or the glycoproteins are present on as branched sugar types. So they both are type of carbohydrates, but here they are in form of glycoproteins and here they are on extracellular side, so here they are called glycolipids. So carbohydrates we have seen in the form of the glyco part shows the carb part. So these are present either as glycoproteins because they are present externally, so they are also known as ectoproteins. And the second type is glycolipids. Glycolipids because they are embedded, so they are also known as phospholipids because they are present on the polar ends of the phospholipids, so they form glycolipids. Endoproteins you can call them. Now, on the basis of discovery, and scientists made different types of models of plasma membrane, which later on found that it was a lipid bilayer. But before that, models of plasma membrane were introduced by different scientists, under which the first model which was given was called as a sandwich model. Now, in sandwich model, it was proposed by Danieli and Dayson. So, Danieli and Dayson in 1935 proposed a model where the plasma membrane was coated on its either side with proteins, and the mutual attraction between the hydrocarbon chains of the lipids and electrostatic force between the protein and the head of lipid were thought to maintain the stability of the membrane. Means the hydrocarbon chains of lipid parts were responsible, and the electrostatic force between protein and the head of lipid they were responsible to maintain the stability. They predicted that the lipid membrane is of size of 6 nanometer in thickness, and each protein layer is of about 1 nanometer thickness, which gives a total each layer, because each layer is present, so total of 8 nanometer plasma membrane they proposed. It is there, lipid bilayer. Then a second scientist named as Robertson in 1960s proposed second model, which was termed as unit membrane model. Unit membrane model. He suggested that a hypothesis was given by him, which suggested that all cellular membrane have an identical trilaminar structure, means a dark, light, dark, 3 types of laminar structure every cellular membrane has. Okay, and then the last, which we today call as the modern model, is third one, a fluid mosaic model was introduced. It was given by 2 scientists, Singer and Nicholson. Who in 1972 suggested that plasma membrane contain a bimolecular layer, both surface of which are interrupted by protein molecules. So this is the most accepted model, which suggested that both the layers of plasma membrane are interrupted by protein molecules, either extrinsic or intrinsic. So protein molecules are there which are interrupting between the bilayer of plasma membrane. Proteins occur in the form of globular molecules, and they are dotted about here, and they is in a mosaic pattern. Means proteins could be anywhere and they are dotted and present in a mosaic pattern. They are either embedded or present outside or present on the surface, anywhere on the membrane. Then on the basis of, because proteins are present here, so when they will enter, when they will go out, on the basis of their movement, some movements were then introduced, like lateral movements of proteins and flip-flop movement. On account of its fluidity and the mosaic arrangement of protein molecules, this model of membrane structure is called the fluid mosaic model. Right? Then, Because cell membrane is allowing some substances to enter or exit, they are supposed to be present some kind of identity markers on it which can recognize what proteins are cell allowing to enter inside them or which protein are supposed to stay outside. So membrane proteins are those proteins which recognize and have certain identity markers on them. So membrane proteins are those proteins which are present on the surface of the membrane. These are responsible for cell-cell recognition, and the proteins which are responsible to recognize are known as cell-cell recognition proteins. Now, carbohydrate chain of glycolipid and glycoprotein serve as the fingerprint of the cell and identify the type of cell. Because we have learned here like glycoprotein and glycolipids here, these because are present on the surface of the membrane, these are responsible to recognize and identify whether the cell is getting introduced by some foreign cell or itself. So glycolipids, carb part of glycolipids, carbohydrate chain of glycolipids and glycoproteins, they are serving as the fingerprint of the cell. They are the fingerprint of the cell as they recognize and identify whether the cell is self or foreign type of cell. Now glycoprotein on extracellular surface serve as the ID tags, means they are acting as ID tags which recognize individual different species or different type of cell. Next membrane proteins, after that we introduce integrins. What are integrins? They provide strength to the cell. Membrane. Strength to the cell membrane. These integrins attached to the cytoskeleton and the extracellular matrix to maintain the cell shape and stabilize proteins. Means, for example, this is the plasma membrane. Here intrinsic protein is present here, and this is providing, it is connected to cytoskeleton. This is cytoplasm, and here is the extracellular surface, extracellular fluid outside of the cell. So this cytoskeleton is connected by this integrin protein, and it is providing stability and flexibility, stability and strength to the cell membrane, so that cell maintain their cell shape. Okay, then we come forward to intercellular junction proteins. Now in intercellular junction proteins, we see these proteins help to cells to stick together to form tissues. Means here two types of cell membrane of two cells are there. So to connect each other, here intercellular junction proteins are present who connect these two cells together and provides a type of adhesion. Means they provide adhesion and stick together through tight junctions and gap junctions. These junctions are of two types. One is tight junction and the next is gap junction. So their primary role is of adhesion. Next, for tight junction, we have seen that they seal cells together into the sheets to provide impermeable barrier to the cell membrane. Means here no substances can enter or go out. So they are basically very tight. So they are tight junctions. For example, intestinal epithelium. Epithelium means the outer layer of our skin. So because of intestine, the intestine outer layer epithelium is made up of tight junction. It prevents transfer of water soluble substances through the cell membrane. Next, gap junction. They provide direct connection between cells, means when two cells are meant to communicate with each other, so these junctions provide direct connection. They are some sort of channels or pores through the membranes of cells and across the intercellular space, means they are pore-like structures which provide communication of cells with each other. So, example is epithelium cells. Next we see different type of receptors are present on the membrane which allow communication or signal transduction, means a signal is sent from one cell to another, then how cells introduce them inside it. So receptors are there. These receptors are type of proteins known as surface proteins or receptors. So they help in signal transduction. So they are known as signal transduction receptor proteins. Now these receptor proteins bind to certain proteins which are also known as hormones. Hormones and other substances on the outside of the cell membrane. That binding process triggers a specific change inside the cell called as signal transduction. Example is insulin, as we know, a hormone which bind to insulin receptors, which causes the cell to put glucose transport proteins into the membrane. So because transport is there, so one more category of proteins is present which are known as transport proteins. So what transport proteins does is they are responsible for passive and active transport, means both type of transport is present, active and passive. Passive transport of proteins means they allow water soluble substances, small polar molecules and ions to pass through the membrane without any ATP cost or energy cost. No ATP is used for these transport proteins to take substances from inside to outside to inside of the cell. No ATP is needed. And the active type of transport proteins require energy because they transport water soluble substances against the concentration gradient. They require ATP because here the concentration is already high and here it is low. So because against the concentration gradient proteins are moving, so here ATP is required. Here proteins are already high, so they are going with the concentration gradient. So till now we have covered in plasma membrane the structural part of it, the composition part, lipids, proteins and carbs, the history part which scientists introduced models, different sandwich model and unit membrane model, and finally the most accepted model, that is the fluid mosaic model. Then we have seen the positioning of different components like carbs part, lipids part and proteins present on the plasma membrane. Lastly, we have seen the type of proteins present on the membrane proteins which are undergoing lateral movements or flip-flop movements. The membrane proteins are already present on the membrane. Integrins are present to maintain communication between extracellular part and the cytoplasmic part, and they are responsible for giving rigidity or cell shape. Then intercellular junction proteins we have seen, wherein we studied about tight junction and gap junctions, which are type of adhesion sticking proteins. Then signal transduction receptor proteins we have seen, where receptor is needed for signal transduction inside the cell. Lastly, we read about transport proteins, which are either active transport protein or passive transport of proteins where ATP is needed or not. So till now we have covered the plasma membrane, out of which questions will be introduced in your competitive exams. Next we will learn about one new other cell organelle. Thank you so much for watching.